Hello, Aditi. Aditi? Hello? Hello? Hello?
We are live, ma'am.
Okay. Aditi? Hello? Okay, good morning all. I'll be introducing today's webinar, Molecular Biology in Food Testing Laboratory. Today's speaker was Miss Anuprita. Expertise in molecular biological techniques in food testing, such as PCR, RT-PCR, ELISA, so on. She has headed the post in Envirocare labs as assistant manager, then quality assurance officer, and now she is assistant manager to technical assistants as Envirocare. Her technical expertise involved molecular biology based analysis and research and development, quality assurance, scientific writing, and designing of project protocols, sales and marketing. In that, she'll be covering branding, trainer for quality assurance as well as for molecular biology. As a assistant manager post, she is handling GMOs, meat specification, pathogen detection, allergen testing, and different other project protocols. So I welcome Anuprita, and you can start with the today's webinar, please.
Thank you. I hope I'm audible.
Yes.
Yeah. So first, I would like to thank the faculty of GN Khalsa College and Envirocare for giving me this very good opportunity to contribute towards shaping the young minds and their development. So really, thank you a lot. So we will be talking about molecular biology and various techniques that are involved in food testing laboratory. So the topics that I'll be covering is really starting from genesis, right from the beginning, that how molecular biology was developed, what are the basic concepts of molecular biology, the central dogma of life, and the various applications in food testing. And these include all the detailed techniques such as GMO screening, pathogen detection, food authenticity testing, and allergen testing. So before we actually venture into the world of molecular biology, I just wanted to share one thought with all of you. That is a very nice quote which I found from Zig Ziglar, who says that if you are not willing to learn, no one can help you. But if you are determined to learn, no one can stop you. So this opportunity that has been given to you all by your college professors is a really good opportunity. Try to grab on as much information as possible. Knowledge never goes waste. So with that, we'll start with molecular biology. So if you look at this image, what exactly comes to your mind? It is evolution, right? So we think about, we immediately go back to Charles Darwin, and he said something, that is his theory of evolution, which says that all organisms on Earth, all life forms have evolved from one single cellular organism. So that is the beauty of nature and science. But we are not looking at evolution today, we are looking at molecular biology. So is molecular biology really also a pre-existing field like medicine or engineering? Not really. If you ask your parents or grandparents, many of them might not even have heard of molecular biology. So this is pretty fresh field, hardly 70-year-old, not as ancient as medicine or other fields that we know. So with that, I will just try to talk about the evolution of molecular biology, how this field has evolved. So it all began with Gregor Mendel.
Anuprita, ma'am.
Yeah.
Yeah, sorry for interrupting, ma'am. Your voice is not clear, ma'am.
Now is it okay? Hello.
Just a minute, ma'am. Just give me a minute. Yes, ma'am. Yes, ma'am.
Hello. Yeah, now it's okay.
Yes.
So as we
Thank you,
talk
ma'am.
about evolution of molecular biology, I will start with the very basic thing. How did it evolve? It evolved from the field studies that Mendel conducted on his field on pea plants. So that he is also known as the father of genetics, and he came up with various laws called as the laws of genetic inheritance. It was his studies that said that there is something that is transferred from one generation to the other. So that was where it all began. And then eventually, over the years, many other discoveries took place over the period of years. So I will not go into the details as to what were the various discoveries. I just mentioned them over here. So ranging from the chromosomal theory of inheritance, the Thomas Morgan's beads on a string theory, then how one gene accounts for one enzyme, and various experiments taken up to show that it is actually the genes which are transferred from one generation to the other. But the major groundbreaking discovery in field of molecular biology was taken up by Watson and Crick in the year 1953. They discovered the double helical structure of DNA. So this was the major groundbreaking discovery which revolutionized molecular biology, and we came to the today's molecular biology, and whatever discoveries happened after this particular event was very quick, and we have come to something that we have developed over a period of years within a very short span of time. It is unimaginable. Something that was in 1953, if you ask them, they would not have been able to think that this also was possible. So then on various other theories came up that how the genetic code is, how it functions, how proteins are developed, how recombinant DNA technology developed. So recombinant DNA technology is a technology that we currently use for producing GM crops, and then eventually moved to sequencing. But again, another major development or discovery that happened after a period of 44 more years was that it was in 1983 where Carey Mullis discovered PCR. PCR stands for polymerase chain reaction. So his discovery was a major breakthrough. And later on, he ventured into Human Genome Project, and further on, many more discoveries took place.
Hello. Can
Hello.
I disturb, please? The
Yes.
voice is not clear, I think. Can you just check?
Now is it
Yeah.
okay?
No,
Hello.
yeah, no. It is not clear yet to listen to.
Is it
Uh,
okay now? Hello.
now it is okay. Yes, yes,
Okay, fine. Now it's fine,
yes,
right?
yes. Now it is fine.
Okay. So this
Okay.
was the evolution of molecular biology, and now that we've seen how molecular biology has evolved over a period of 50 years, let us come to the very basic question that I'm sure most of you have got in your mind: that what exactly is molecular biology? So it is very difficult to explain to a layman that what exactly this complex field is. But to put it in simple terms, it is a branch of science which concerns the study of biological activity that happens at a molecular level. So if we look at all the various life sciences or biological studies, it typically involves studying the cells, organelles, or other mechanisms that happen. But in molecular biology, we look at the various biomolecules which work at a very ultra microscopic level, deep into the nucleus where the secrets of life lie. So, molecular biology involves the study of the genes, DNA, RNA, and proteins. So, these are the molecules that are studied in this field. So, what are the various terminologies that are, these are some of the buzzwords that we hear commonly in molecular biology. So, gene, DNA, RNA, protein. I'm sure many of you have heard these terms, but very few people understand what exactly they mean. So gene is it is a sequence of information which acts as a basic physical and functional unit of heredity, and it is the genes that are actually transferred from one generation to the other. Now, DNA, RNA, and proteins are something that we hear very commonly, but in chemical terms, what is exactly a DNA? So, DNA stands for deoxyribonucleic acid. It is a genetic material of most organisms rather. Even you humans also, it is a genetic material. The RNA, on the other hand, stands for ribonucleic acid. It is a genetic material in the lower forms. Like, for example, the recent coronavirus that is going on, its genetic material is an RNA. But in humans, what role does an RNA play? It plays a main role in gene expression. And proteins, we all know they are very important they are called as the building blocks of life. So proteins are these large biomolecules made of long chains of amino acids. We have 20 amino acids, and proteins are made out of the combination of these 20 amino acids. And they play a very significant role in our functioning of our body. Now, there is a very interesting phenomenon that takes place within these three molecules, that is DNA, RNA, and proteins. So this phenomena is called as a central dogma of life. Before we go into the central dogma of life, I've just depicted how the DNA structure is and how the RNA looks like. So I'll not go into much of the detail. I'm sure you might have studied it somewhere. So what is the central dogma of life? Central dogma of life is a basic principle on which our entire existence depends. So if you look at this image, you can see that within the nucleus, there is a DNA. So DNA is a double-stranded sequence, and then it is converted into an RNA. This process is called as transcription. And the second step that takes place in the central dogma is called as the translation, wherein the RNA is converted into the proteins. So first, before all this happens, DNA has to replicate. And in the S phase of the cell cycle, the DNA goes on replicating. One double strand of DNA forms two forms four, and so on. Exponentially, DNA goes on replicating with the use of certain enzymes in the nucleus. So these enzymes typically involve DNA polymerase, helicase, and certain other proteins also. So it's a very complicated process, but based on this particular process, the DNA goes on replicating during the cell cycle. Once the DNA replicates, the next step is transcription, wherein it is converted into a messenger RNA. This process also takes place within the nucleus, wherein the DNA is converted into an mRNA, but with the help of the enzyme RNA polymerase, and also certain transcription factors are involved in this process. The mRNA then moves from the nucleus into the cytoplasm of the cell, wherein it gets converted into protein. But how does it happen? It takes the help of something called as a ribosome. So I'm sure in cell biology you might have heard or you might be aware that there is something called as rough endoplasmic reticulum. So on this rough endoplasmic reticulum, there are these ribosomes. So ribosome along with the mRNA start decoding this instruction that are present on the DNA, and it gets converted into chain of amino acids, which eventually folds and forms the functional protein. This is the central dogma of life. So I've just shown this animation where it is showing how an DNA is converted into an mRNA, and later with the help of ribosome, how the tRNA helps in formation of the chain of amino acids, which eventually will form the functional protein. This is the central dogma of life. Now that we've seen the basic principles of molecular biology, what are the various techniques that are developed on the basis of these molecules? So broadly, we can categorize it into DNA-based methods, RNA-based methods, and protein-based methods. So there are four techniques involved under the DNA-based methods, which are called as recombinant DNA technology. So recombinant DNA technology, where exactly is it used? It is used most commonly in creation of these GM crops. I'm sure you might have heard of BT cotton or BT brinjal. So these crops are developed with the help of this technology called as a recombinant DNA technology. I will be explaining recombinant DNA technology more in detail further on, but here I'm just introducing these terms. So PCR, the second method is called as PCR. What exactly is PCR? PCR stands for polymerase chain reaction. And based on this technique, like I mentioned, it was Carey Mullis who discovered PCR. And based on this PCR, it has undergone evolution over a period of years. And a lot has been done by the use of this PCR technique. So that also I will be explaining more in detail further. Then there is something called as southern blotting. So southern blotting is a technique which is used to differentiate between different types of DNA, wherein the DNA is cut with the use of various enzymes and with the help of gel electrophoresis, the DNA, different types of DNA are identified. This technique is typically used in DNA fingerprinting, paternity test, and all these techniques use the southern blotting. And then the last technique is microarray. So microarray is a very evolved technique. It usually consists of the where the entire genome is taken into account, and we can study how different genes are expressed at different stages of the cell cycle. So you can use different cells at various stages, and you can extract the genome, and we can find out which genes are being overexpressed or which genes are being underexpressed. So this is a very evolved technique in molecular biology. The second type that I mentioned are based on RNA. So RNA, the technique that is used is northern blotting. So what exactly is northern blotting? Northern blotting and southern blotting both function on the same term, wherein the like I mentioned in southern blotting, the DNA is cut using various enzymes. Here in northern blotting, what happens is instead of DNA, we use the RNA, and various enzymes are used to cut the sequence of RNA, and then we can distinguish between the different types of RNA. Again, gel electrophoresis is also used over here, and the last stage involves the use of radiolabeled dyes, which helps us in identification and comparisons. Okay, so this is northern blotting. Then the third and the last type of molecular broad categorization involves protein-based methods. So basic concept of any protein-based methods is that interaction between antigens and antibodies. So these two molecules are complementary, and then they form a complex called as antigen-antibody complex. So these three methods, that is strip test-based method, Western blotting, and ELISA, they all dependent on the same basic principle, that is antigen-antibody interaction. So strip test is something that is typically it consists of a strip wherein the antibodies are immobilized onto these strips, and the strip is dipped into a solution that contains your sample, and then you can detect whether the particular protein of interest is there or not. This technique is most commonly used in a pregnancy test or for detection of various contaminants. We can use a strip test, and it is usually used on field because it is a very rapid technique, and quickly it can detect on a qualitative basis whether the molecule that you're looking for, that particular protein, is present or not. Then there is Western blotting. Western blotting again has the same principle or concept like the Northern blotting or Southern blotting. Only difference is here, instead of going for the DNA or RNA, we are studying the proteins and the polypeptides. Then we come to ELISA. So ELISA is something that I'm very sure everyone has heard of at some point of time. It is a technique most commonly used in the medical field for detection of various diseases and quick detection, diagnostics especially it is used. So ELISA stands for enzyme linked immunosorbent assay, and this also depends on antigen antibody interactions. It has various applications in food testing also, which I will be going into more detail further. So like I mentioned, first we'll be studying the recombinant DNA technology. So if you look at this image depicts the various combinations and manipulations that we do to the genetic material in order to create various different types of crops. Like for example, like I mentioned earlier, the BT crops. So these crops have some modification done to them. Why do we do it? Basically the intention is very good, that we want to develop crops which can sustain these crops can become resistant to certain strenuous conditions, like for example they have resistance to pests or insects, or sometimes to high salinity. So these soils should be able to sustain those severe conditions, which is why this technology has been evolved. So what exactly is recombinant DNA technology? The technology involves alteration of the genetic material. So the DNA in question is altered in order, wherein what we do is we cut a particular strand of DNA and we insert our gene of interest, so that in order to create a combination of DNA called as a recombinant DNA. So I have just shown what are the tools that we use in this technology. So we need two enzymes, two types of enzymes. One is restriction enzymes. These are also called as molecular scissors. And then the second type of enzyme we use is called as a ligase. So this act like the restriction enzymes act like a scissor, and the ligase acts like a glue. So here in this image, I'm just showing how the recombinant DNA is created. So there's something called as a vector or a plasmid. So this circular DNA that you can see is a plasmid, and it is typically found very commonly in bacteria, and they have a very high ability of transforming, that is getting transformed from one bacteria to the other. So these gets replicated very quickly, and then we get a large quantity in a very quick manner, which is why the plasmid has been used in recombinant DNA technology. So what we do is we cut a particular sequence of the plasmid DNA and we insert our gene of interest. So in case of GM crops, the most common gene that is used is a cry-gene, which is responsible for insect resistance. So that gene, cry-gene, is incorporated into the plasmid, and then so this process of cutting is done by the restriction enzymes, and then it is pasted with the help of ligase, and then we get something called as a recombinant DNA. So this recombinant DNA, once it has been created, is again put back into the bacteria, and it is allowed to replicate in order to produce a large amount of the recombinant DNA, as well as the expression of a large amount of the final end product, that is your protein of interest. Like for example, I mentioned over here the cryogens. That is how the recombinant DNA technology works. And the second most common technique used in molecular biology, as I mentioned, was PCR. So, what does PCR stand for? PCR stands for polymerase chain reaction. What is the purpose of PCR? Many people ask this, that why exactly are you using PCR? The most basic principle of PCR and the main purpose why we use PCR is to amplify the DNA. So whenever we get different kinds of cells, it could be plant cell or an animal cell, the DNA that these cells contain is very small in quantity. So there has to be something, how do we increase the quantity of this DNA so that we can do any research further on or use this DNA? So that process, the amplification part, is done by the PCR technique, which was developed by Carimelis. And this is the first step of various research activities. So PCR is a very basic step in the molecular biology. So I have just to simplify this concept, I have developed an analogy. So imagine if you are baking a cake, you need some ingredients, and then you need in order to bake the cake, you need an equipment also. So that equipment in this case for PCR is the thermal cycler, and your ingredients are these are the PCR components which become your ingredients. So what ingredients do you need in order for a PCR reaction to happen? You need the DNA sample that you are extracting from your product. That it could be a food sample or it could be a leaf sample or anything. So when you are extracting the DNA, this becomes your sample. Then you need something called as a primer. So what are primers? Primers are oligonucleotide short sequences which are complementary to your DNA sample, and they will recognize a specific sequence in your DNA that you want to amplify. That is the objective of a primer. Then you need something called as nucleotides. So there are four basic nucleotides in a DNA, that is cytosine, guanine, adenine, and thymine. So ATGC. This is something you might have heard. So these are one of the most important ingredients, because this has to be added to your DNA strand. And then there is something called as Taq polymerase. So what is Taq polymerase? It is a very important enzyme. The term Taq comes from Thermus aquaticus. This microorganism is able to sustain very high temperatures, which is precisely what we do in PCR. So this thermal cycler, the entire concept of PCR works on the principle of temperature variations. We go from very high temperature to medium temperature to very low temperature, or there are various cycles which includes the temperature fluctuation. So the enzyme that has to function has to be such that it can sustain all these temperature variations, which is why we use something called as Taq polymerase. And then there are buffers, and we need PCR tubes also to carry out a reaction. So once the ingredients are together, we put it into the thermal cycler, and then the PCR happens. But what happens? What are the basic principle of PCR? There are three steps involved in a PCR cycle. So first step is called as denaturation, the second step is called as annealing, and the third step is called as extension. So what exactly happens is, once you start a reaction, it will always be at a very high temperature, at 94 to 95 degrees. The double-stranded DNA is a very complex molecule, and it is very strongly bound to each other. In order to break these bonds, you have to use a very high temperature. So once we elevate the temperature to 95 degrees, the two strands separate. And then at 55 degrees, our primers that we had added, which are complementary to the forward and reverse position, will go and bind to the complementary sequences in the DNA. Then at 72 degrees, Taq polymerase will start adding DNTPs, that is these nucleotides. It will go on adding complementary sequences to the DNA, which is how one copy of your DNA forms two. And this process we can set, just like in your oven, you can set the temperature and for how long you want to keep it. In a PCR also, you can set the temperature fluctuations as well as the time for which you want to run it, the number of cycles that this process goes on repeating. Typically in PCR, we go for 30 to 40 cycles. So what happens is, when you had a one copy of DNA, by the end of 30 cycles, you will get a billion copies of this DNA. So that is the beauty of this technique. And that is why this was the most groundbreaking discovery in the history of molecular biology. So then in this animation, I've just shown how the two strands separate at 94, how the primers will anneal, and how the Taq polymerase will go on adding the DNTPs, and how we eventually get the copies of DNA in PCR. Now, what I had earlier explained was the very basic form of PCR. What technique we usually use in lab is something called as real-time PCR, or RT-PCR, as it is commonly known as. So what is the difference between PCR and an RT-PCR is that in real-time PCR, you can actually quantify the DNA. In PCR, it happens only at the end of the cycle, whereas in real-time PCR, you get a graph in real time. As the reaction progresses, you will get a graph. So what is the difference basically is that the three steps are common, that is denaturation, annealing, and extension. But there is one additional step wherein you use a probe. So along with the primers, you will also add something called as a probe. So typically in RT-PCR, we use something called as cyber green. So what this probe consists of, it is a dye. So the dye will have, so this sequence of DNA will have a dye attached. It has a reporter dye and a quencher dye. So the concept of that is used in real-time PCR is called as FRET, that is fluorescence resonance energy transfer. This is the concept. So the reporter dye, it goes on emitting a fluorescence, and the quencher dye goes on quenching it. So this is balanced when the sequence is intact. So the first step is same as PCR, wherein we use 94 degrees centigrade to break down the strands. The two templates, they separate. Then we add the probe. The probe is complementary to some known sequence in your DNA of interest. So it will come and bind to the DNA, and then that is called as probe annealing. Then what happens is that you add your primers. So primers, again, as usual, they get annealed, and then your Taq polymerase will start extending the sequence by adding DNTPs. But here one major thing happens is, when your Taq polymerase reaches this probe, it is going to break the probe. So the reporter and the quencher, they get separated. And now the quenching activity does not happen, because of which the reporter dye goes on emitting your fluorescence, and you can see that is this signal is being emitted. so that my DNA has been amplified. And as the number of copies go on increasing, but obviously the signal also goes on increasing. So if we are screening for anything in molecular biology, like in our laboratory, when we screen for anything, if you are checking for some contaminant and we are looking for a particular sequence, if we are getting a signal, it means that the DNA of interest is present in the sample. That is the basic difference between PCR and the RT-PCR, and this is the principle how it works. Again, here I've just shown a animation which just shows how the Taq polymerase breaks the probe, and then the reporter and the quencher is separated, and we'll get a fluorescent signal. So this reporter dye is going to emit your signal. Now that we have seen that how PCR and RT-PCR work, so how this technique has evolved over a period of years. The first principle which I explained was PCR, that is polymerase chain reaction. This is used for amplification of DNA. This is a very basic step, like I mentioned. But there are some challenges when you use PCR. First thing is extremely time consuming. You need to do post processing. That is anything that you run, any reaction that you run in a PCR, you have to run a gel electrophoresis. You have to do further studies on that. So which is a major challenge with regards to PCR. It is also doesn't have a very good sensitivity or a resolution. Also, if you repeat the experiment, you really can't say that okay, I'm going to get the same results. So which is why PCR is not very reliable, and we go for RT-PCR or qPCR in most of the cases. This PCR is typically used in research laboratories, whereas RT-PCR is used when you want to quantify anything and when you want to be really sure. You should definitely go for real-time PCR. So real-time PCR is also called as qPCR, that is quantitative PCR. Its basic function is for quantifying the DNA. Here, what are the advantages that you have with regard as compared to PCR is that there is no post-processing. You don't have to run an agarose electrophoresis. You don't have to separate the DNA. You get the detection in real time. Like I mentioned, as the reporter dye is separated from your quencher, the signal goes on getting amplified. And as you get more and more copies of DNA, the signal is going to increase. And you will get a graph as the reaction progresses. You don't have to wait after your entire reaction to say, okay, whether my DNA is present or not in that. This is the benefit that this technique offers. It is highly sensitive and it has a very good resolution. You can make out the differences between minor differences in DNA also in this technique. Now, something very recently what has evolved is something called as dPCR. I don't know whether you're aware or not, but dPCR stands for digital PCR. Now, what is the principle and everything is same as RT-PCR. Here also we are going to use probes and dyes and everything. The only difference is that how we run the samples. So in dPCR, what happens is we are going to split the sample into different wells, and into multiple wells, whatever sample has been fractionated, you can ensure that every DNA is taken into account, and which is why this is a very highly sensitive and more accurate technique. It is more reproducible, and you can do multiple. You can check for multiple types of samples and multiple DNA also in this technique. This is how the technique of PCR has evolved over the years. Now let us look at ELISA. So like I mentioned, ELISA stands for enzyme-linked immunosorbent assay. So what exactly does it mean is that if you can see this well plate, this is where these are the polystyrene plates wherein you can run a ELISA reaction. What is the principle behind it is, again, like I mentioned earlier, it is about antigen and antibodies interaction. So specific antigen antibody reactions will take place and it will form a chromogenic product. At the end, what happens is that a chromogenic product. So this blue color will turn into yellow, and that's how you can detect it via a spectrophotometer or something called as an ELISA reader. I'll go into more details. So there are four types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and comparative ELISA. So direct ELISA is very basic. Your antigen is immobilized onto the plate. We add our primary antibody, which is going to be complementary to our antigen of interest. And then this antibody is complex to an enzyme. Usually we use horseradish peroxidase or alkaline phosphatase. These are the two enzymes that are typically used. And then we add a substrate, and the enzyme will convert the substrate into a colored product, which will eventually give you a reading. That is, you can quantify. So this is a very basic technique of ELISA, and it is direct. That is, you use only one antigen and one antibody. So then there is second thing called as indirect ELISA. So here the detection takes place in two steps. The antigen like direct ELISA is also coated onto the well, but we use some mediator called as a primary antibody. So primary antibody is complementary to your antigen. It will bind specifically to your antigen, and then we add a secondary antibody in this case. So secondary antibody is complex to the enzyme, and then it is going to convert your substrate into a colored product. This is more specific and sensitive. The addition of an intermediate will ensure that nothing gets missed out. You don't get any false positives or anything. So that's the difference between direct ELISA and indirect ELISA. What happens in sandwich ELISA is that there are three antibodies involved. So your plate of the immobilized plate that I've shown in this image, here instead of antigens, we are going to add antibody. That is called as a capture antibody coated plate. So then we add a sample which contains the complementary antigen, and it will form a complex. Then we use the detection antibody. This antibody also has an epitope which is specific for the antigen, and then we add a secondary antibody which will bind to your detection antibody. So this is, it kind of forms like a sandwich, one layer above the other. So that is why it is called as a sandwich ELISA. And here, this is a very good technique, and most of the detection kits that we use are based on sandwich ELISA, because it has a you can be sure that the results will be accurate when you go for this technique. And here, the detection is it's a direct proportionality. So if your sample contains a high amount of your antigen, it is going to give you that much amount of signal on the spectrophotometer. The fourth type of ELISA is something called as comparative ELISA. Here what happens is that you are again going to use capture antibodies, but instead of adding just your sample, you are going to additionally incorporate something called as enzyme conjugated recombinant analyte. So you are purposely introducing a competition to your sample, and these two antigens will compete with each other to bind to your antibody. So based on so since you're going to add a fixed amount of this second antibody, that is enzyme conjugated recombinant analyte, we will come to know what is the concentration of the enzyme or the protein in our sample. So based on that, we can detect the presence of proteins. Now we come to something called as the application of these techniques in food testing. So food is a basic necessity of life. As we all know, we can survive without most things, but we cannot survive without food. I'm sure in this lockdown period, everybody just is dying to get out and when they will have good food or hotel food or something like that. So, but it is also equally important that we assure the quality of food, and which is why it is extremely essential that we screen it. So, what is the applications in food testing? So there are different ways in which we can do that. We have different applications. One is in GMO screening. We go for pathogen screening, then authenticity testing, and screening of allergens. So before I go into deep into this particular aspect, I will just say that it is extremely important that we screen the food that reaches the market. Why? Because we have to assure the quality of the food that we are delivering to our customers. And there are various national and international bodies such as the Food Safety and Standards Authority of India, the European Union, then the FSANS, or even the New Zealand standards also. There are various standards which say that okay, this particular food has to comply to these particular requirements, regulatory requirements. So in order to ensure that this happens, we have to test the food, which is why it is very much important. And then there is a third concept called as surveillance. So this activity of screening food isn't a one-time activity that okay, I've developed my product, is it meeting these standards? This is not a one-time activity. It has to be done on a continual basis, because even one flaw in your product can affect a person who is consuming it. So that is why it is very much critical. So what is GMO screening, or why do we need to screen for GMOs? Before we go into that, what exactly is a GMO? So GMO stands for genetically modified organism. Like I mentioned earlier, what is a genetically modified organism? It is an organism in which we are altering the DNA, and the modified crops such as cotton or BT brinjal. These various types of crops are genetically modified, and the various countries, they don't really have. Not everyone has approved GMOs. There's a long going debate that has been saying that whether the GMOs are good or bad. There's a certain fraction of people who say, okay, GMOs have these potential abilities and they are good for us. But there is another fraction of people who always say that okay, there might be some negative impacts of this GMO too. For example, development of superbugs or resistant strains of pesticides. So these can be some of the negative impacts of GMO, and it is a long going debate, which is why not all countries are open to GMOs, and especially when you're trying to claim that a food product is free of GMO, you have to get it tested. So this is GMO screening. Then the second thing is pathogen screening. So typically, the common microbiological techniques also can be used for screening of pathogens. So food, like we all know, may sometimes contain these harmful organisms such as E. coli or Listeria or Salmonella. And these, even in small quantities, if it is consumed by a person, can have severe impacts on their health. Which is why detection at an earlier stage, if your product is contaminated by these pathogens, it has to be done at an earlier stage. And that is why pathogen screening is very much important. The third most commonly common techniques that we use is something called as authenticity testing. So, what is food authenticity testing? So, basically, authenticity is that is your product actually what it is? So many a times there are a lot of adulterants that are added to your product. For example, I've just shown meat. So meat, now if you think that it is chicken, is it really chicken? Many a times it is adulterated by some other kind of meat in order to lower the cost. So this is why we have to ensure that okay, what is being delivered to your customer is actually authentic. And that is where authenticity testing comes into picture. Then food also has something called as allergens. So not everybody, but it is a studies which says that 1 in every 10 people have allergy to some different type of food. So most common allergens that we find in different types of food are in dairy, gluten, seafood, then soya allergen, egg, peanuts, shellfish, and peanuts. So these are the most commonly identified 8 allergens. So people, not everybody, but some of the people are allergic to these particular the proteins that are associated with these food products. So which is why we need to screen our food products and also display it on the labels. So there are various regulations on this which says that on the packet of food, the if the product contains allergen, it has to be declared, which is why it needs to be screened. So now we will just see these techniques in detail. So since during this lockdown period we cannot have you all here in the laboratory, I just thought we'll bring our molecular biology laboratory at Envirocare to you. So this is how our molecular biology lab looks like. So what is a process flow? In any molecular biology laboratory, it is extremely vital that there is a unidirectional flow of your products. Why? Because it has to be ensured that there is no cross contamination. So there has to be a unidirectional flow only. So what is the process flow is once we receive any sample of food, we will weigh it in this sample receipt area when the sample is weighed and it is prepared. This sample is then transferred via aseptic means, that is via a pass box, into the DNA extraction room. So this second room, what we are seeing is the DNA extraction room, wherein our scientists and analysts, they will extract the DNA from the product by using various extraction enzymes. Then the extracted DNA is again aseptically transferred to the master mix room. So what happens over here is that the DNA is added to a master mix, and this is processed further to the PCR, RT-PCR room, wherein the sample is processed for the analysis and we can do the amplification of DNA. So this is the process flow of the molecular biology laboratory. Now, the first technique that I will be explaining in detail is GMO screening. So before we go into what exactly is GMO screening and how we do it in the lab, we'll come to what is a GMO. So GMO stands for genetically modified organism, and I've just shown the different types of most commonly altered types of GMO that are present. That is GM cotton, then there are GM maize, and GM brinjal. These are some of the most common types of GMOs. So the technique that is used to prepare these genetically modified organisms or GM crops is called as recombinant DNA technology, which I've already explained earlier. So the crops are PT crops. And the screening, why should we screen for these organisms is because it is a regulatory requirement. Whenever you try to claim that a product is organic, it should ideally not contain anything foreign to it. When you are altering the genome, then you are introducing something foreign that is not its authentic gene. That is why there is a discrepancy, or there is a concept which says that okay, GMOs are good or bad. Like I mentioned, there is a long going debate about it, which is why we need to screen our products. And also another reason why we need to screen GMOs is because various regulatory bodies all across the globe have set different labeling thresholds. If your, for example, EU has set a threshold of 0.9%. So if your food product contains 0.9% also of an ingredient which has been derived from a GMO, then it has to be put on the label. So such labeling thresholds or such stringent thresholds in the range of 0.9 to 3% have been set for GMOs. And that is why we need to screen for it. So most commonly, what do we screen for in these crops is these two events called as CAME 35S and TNOS. Now, what is CAME 35S and TNOS? So this is a promoter agent. CAME 35S is cauliflower mosaic virus, and it is 35S is the region that we are screening for, and it is a promoter sequence which is present in the gene, and it is the most common event that is used in while preparing a gene, which is why we target these two particular things. The second one is a terminator sequence. So a promoter and a terminator, that is most common in GM crops. That is why these two events are screened. What is the process flow when you are screening GMOs? So whenever a sample of food, like most commonly we receive corn and corn-based products, rice-based products or soya products, and also cotton in various forms such as seeds, it could be leaves, raw cotton or fibers also. So once the sample is received, the first step is DNA extraction, with which I'll be going more into detail. I'll explain each of these process in detail further. So first the DNA is extracted. Then we prepare something called as a master mix. So master mix includes your ingredients for the PCR reaction. And then the DNA is incorporated into this master mix and we amplify the DNA. So we let the cycles of DNA happen and we amplify the DNA. And based on the graph that we get on an RT-PCR, we will interpret the results and the report is released from the laboratory. So what exactly, how we go ahead about with regards to DNA extraction and how we do the quality check. So first, I've just shown these are the cotton seeds. So the sample, a known quantity of the sample is weighed. Then it is subjected to lysis with the help of a mortar pestle and then using a DNA extraction buffer. This buffer typically contains agents such as EDTA or SDS, which will help in the lysis of the cells. Once the lysis is done, the solution is transferred into a centrifuge tube and we centrifuge it. So this is a centrifugation machine wherein we can set the temperature and time, and we will centrifuge the sample. Now, in order to get a pure quality DNA, first we need to remove all contaminants that are present. So what could possibly be present as a contaminant is the proteins or RNAs. So we need to remove the proteins and RNA that are usually present in the cell. How do we do it? We add some enzymes called as RNases and proteinases. So RNase is an enzyme which is going to break down the strands of RNA, and proteinase is an enzyme which is going to break down any proteins that are present. And centrifugation will help in separation of these layers, on the basis of which all these fragments that have been generated, they will go in as a pellet, and our DNA will remain in the aqueous layer. So we will collect the supernatant. The next step involves organic extraction. So different types of organic solvents such as phenol chloroform or phenol chloroform isoamyl alcohol. These are the various combinations of organic solvents that are used. And again, your supernatant is subjected to, after addition of these solutions, we will subject it to centrifugation. So the DNA will always be in the aqueous layer, and rest of the contaminants like lipids and anything else that is remaining will go into the organic layer. The last step of DNA extraction includes ethanol precipitation. So chilled ethanol is added and it will be used to precipitate the DNA into the out of the solution. Now, once we have an extracted DNA, we have to check for its quality. Before we go ahead with the RT-PCR reaction, first thing that has to be done and the most vital part is assuring that it is a pure quality DNA. How do we do it? Is that we have a machine called as nanospectrometer. On this, you can get the ratio of absorbance, that is A260 by 280 ratio, which will tell us whether the DNA is good quality or not. Typically, the ratio should be close to 1.8. If you have a ratio of 1.8, it means that it is a good quality DNA. And only if the ratio is between 1.6 to 1.8, we go ahead with the further steps. Otherwise, extraction has to be repeated. Then the second step is master mix preparation. So what happens over here is once you've extracted the DNA, we need to add the other ingredients of the RT-PCR reaction. So typically in the market, various screening kits are available for GMO screening. So what is the composition of these kits is like I mentioned in the principle, we need a probe. So cyber green dye is our probe. Then the enzyme Taq polymerase, buffer, and the primers, as well as we have a positive control. So these are the ingredients that are added to a PCR master mix. Then our DNA which we have extracted is added. So cyber green dye plus polymerase plus our primers, and also there is a positive control. This along with our sample DNA will form our final reaction mix, and typically it is 20 to 25 microliter reaction volume which is added to the PCR tubes in a particular sequence. So positive control, so instead of your sample you will add a positive control. And there is also something called as an endogenous gene. So what is an endogenous gene? Before we move ahead with the analysis of samples, first we have to assure that whatever DNA we have extracted from whichever matrix, is it authentic to the matrix? For example, if we are using a sample of cotton, we first have to assure that okay, the DNA I have extracted is actually from cotton or not. Which is why we will add endogenous gene, a primer which is specific for an endogenous gene which is present in cotton. So before we add our sample DNA, we will add this particular primer. Instead of adding these primers for terminator and promoter, we will use a primer specific for endogenous gene and we will amplify the DNA. Only if it is giving a positive detection, then we will go ahead, okay, that this is the extraction process was correct and the DNA which I have obtained is actually from the matrix. Then we move ahead with the samples. After the extraction and master mix and the amplification process, this is the third and the most important step of a real-time PCR. So that the mixture that was prepared earlier is now placed into the real-time PCR machine. And then here we have a lot of options wherein we can set the protocols as to what temperatures and what time combinations the process has to be done. Typically, it takes 1.5 to 2 hours. And based on the fluorescence emissions, at the end, we can determine the CT values. And this CT values is based on these CT values, we will interpret whether the sample contains GMO or not. So interpretation of results. So what is CT value? CT value of a reaction is defined as a cycle number when it is a cycle number where your PCR product is going to start showing a signal against the background signal. So if you look at these graphs, this is the most important, these two are the important ones. So what exactly does it mean is that this flat line states that this is a background noise and it forms a threshold limit. So any curve above this is a fluorescent. These are the fluorescent emissions. And the CT value, so if I consider this particular curve, if we are getting a signal at this particular curve, this means at this cycle number 20, we are getting a peak. So based on these CT values, we will interpret whether the product contains a GMO or not. It is very clear in this graph. So any peak above more than 4,20,000 people died, and this is because of foodborne illnesses or pathogens. So, timely detection of these pathogens in these food is extremely critical. Though food business operators take various measures, I'm sure they do a lot of things in order to ensure that there is no pathogen in the final product, but still sometimes it may happen that your product contains some particular pathogen. So typically, so that is why it is very important that this pathogen this food product which is having this pathogen doesn't reach a consumer and doesn't have any health implication. So this is why we need to test for pathogens. So typically, microbiological techniques are available for screening of pathogens such as E. coli, Salmonella, or Listeria. But the challenge is that it takes 3 to 5 days, and this is a big challenge when, especially when food products are exported. So by the time it reaches a consumer, you cannot do anything. So that is why it is essential that we go for rapid techniques such as RT-PCR based techniques. So these techniques will give us results within 24 to 48 hours. And we can take, and the food business operators based on these results can take some actions. If they want to recall the particular batch which had this particular pathogen, they can do it, which is why we need to go for this technique. So these pathogens such as E. coli, Salmonella, or Listeria. So E. coli and Salmonella are typically found in meat and poultry products and dairy products, whereas Listeria is commonly found in frozen products. Now, how do we screen it using rapid techniques? We will use the kits. So there are various kits available in the market, and what happens is just like GMO screening, instead of us screening for a particular promoter or terminator sequence in a crop, we are going to screen for a sequence which is specific for the particular microorganism or at a species level. So that is how we will amplify the DNA and we can say that okay, this particular product is free of this particular pathogen or not. Typically, pathogen shouldn't be present in any food product. So the process flow over here is that the first thing that we do is the food product when you receive it. First step for any pathogen screening is we need to enrich the product. That is, the DNA that you are going to extract from the food sample will be very small. So we need to give, if at all it has even one colony, in order to detect that, we need to give it sufficient time to grow so that we have sufficient amount of DNA so that we can proceed with the PCR reaction. So first step is enrichment, wherein we allow the microorganism to grow. Then we extract the DNA. Then as per the typical RT-PCR reaction, a master mix is prepared based on the kits and based on the specific primers, and then the DNA is amplified and result interpretation is done. So I've just tried to show how exactly we proceed with the samples. So first the sample is weighed. Then we are going to enrich it, like I mentioned. We need to increase the quantity of the pathogen if at all it is present. So we allow it to be enriched in the BPW buffer at 37 degrees centigrade. It is pre-warmed. So the timing is typically in the range of 18 to 20 hours. And for meat product, it may be less. So based on that, we allow it to enrich. And the next day after enrichment, we will collect the enriched sample. And this enriched sample is centrifuged again to get a layer. Then the kits usually contain something called as a lysis buffer. So in order to lyse the cells of this particular pathogens, and then we can get extract the DNA. So the lysis buffer is added and it is vortex. Then heat treatment is done by placing it onto the heating block at 95 degrees. Again, the particular vials are again centrifuged and then the DNA is extracted. So this is how the food sample is processed with regards to pathogen detection. First is enrichment, then you centrifuge it, addition of lysis buffer to break down the cells, and the DNA is extracted. Second step, as usual, is amplification and interpretation. So the kits will have specific probes, primers, as well as buffers. So whatever DNA we have extracted, we will add to this particular master mix and we will run the reaction. Along with the samples, we also add a positive and a negative control to be doubly sure. And again, like I showed the graphs for the PCR in GMO screening, same way CT values, on basis of CT values, we can make an interpretation. Now, the next concept is called as meat speciation. So authenticity self-testing is extremely important. Like I gave you an example, many a times it so happens that for economic reasons, the food can be adulterated with different kind of meat. Especially these are the most typically, these kind of alterations are done like chicken, dog, cow meat, like this bovine meat, then sheep, horse or pig. So you may never really know what you are consuming. So it is extremely important that the meat speciation also be done. This is extremely critical because it has economic implications, implications of public health, and it is a religious matter for some as to what exactly they are consuming. Also, nowadays people are extremely aware about all these things. So as food business operators or anyone who is involved into the food industry, it is extremely important that if you are involved with such kind of product, the the meat speciation is also critical for halal testing. It is one of the requirements. How do we go ahead with meat speciation? Again, there are some kits in the market which you can use for categorizing or screening or differentiating between different kinds of meat. So the kits usually contain the primers. Now this is based on COX gene. So COX gene is something that has a very high, it is used as a molecular marker. So it can differentiate between very small or minute mutations as well at the species level, which is why this particular sequences are used. Again, we will use a probe that is cybergreen based probes, and of course, while running the sample, a positive and a negative control is also added. The first step, so I've just shown a flow as to how the meat speciation is done. The samples are weighed, DNA is extracted again by the same method what we are using, PCI based method. Earlier, I've already explained it. Then we establish the concentration, whether we are having a sufficient concentration or not. Usually, the concentration of DNA that is taken is between 90 to 140 nanogram per microliter. This is the concentration that is taken. Then further on, the master mix preparation is done, and we subject it to the PCR reaction. Based on the graph, we can conclude. If now this graph is showing that okay, it is having a very high peak, it shows that a particular product is having a particular sample or a primer that you are particularly screening for. That was about meat speciation, wherein you can differentiate between different types of meat and ensure the authenticity. Now, coming to food allergens. So, what exactly is an allergen? So, many people, like I mentioned earlier, also that many types of food contain certain protein to which people are allergic. So, 1 in every 10 people are allergic to some kind of food. Typically, these are like egg allergen. There are eight, like I mentioned earlier, there are 8 types of allergen, and these protein, they what how do they do cause an allergy is that these protein elicit an abnormal immunological response in some people. So it usually presents in the form of mild rashes, and that is nothing. Sometimes it is very mild, but sometimes it can have severe implications like swollen airways or difficulty breathing. And in the worst case scenario, it can cause anaphylactic shock and to lead to the death of the individual. So it is very important that these products be screened and we go for allergen testing. There are many laws also, even in FSSAI in the labeling declaration, they have mentioned that you need to declare the allergens if at all it is present in your product. So that when the consumers read the food label, they're aware that whether what am I consuming. So if a particular person is allergic, then he can take a call whether to consume the product or not. That is why it is very important. So allergen testing, the process flow, how we go about is that whether it is a solid sample or a liquid sample, the sample has to be subjected to homogenization and then extraction. Then we run the ELISA test and the results are interpreted. So how exactly we go about with sample preparation? Usually ready-to-eat products. I've just given an example for ready-to-eat product. So first thing, it is going to be homogenized. Then the sample is weighed. A dilution buffer or extraction buffer is available. Now typically it is in 10 folds or 15 folds. This buffer needs to be diluted. Then it needs the sample and the buffer needs to be mixed properly, vortex properly, and then it is incubated. After incubation, this product sample is centrifuged in order to extract the particular protein into the supernatant, and we collect the supernatant. So, how do we run the reaction after extraction? Is that we place the wells into the microplate holder. So, whatever number of reactions we want to do, accordingly we will place the wells. The standard as well as sample is added into these wells. Incubation is done. Then, in order to ensure that excess antibodies, whatever we add into the reaction, they are drained off. Then, frequently we will do washing with the buffers, and after the washing, the excess is drained off. So enzyme and conjugate and incubation is done. That is the next step. That after washing and all, we will add the enzyme, which is going to give you a colored product. So again, frequent washing is done in order to ensure that any additional or any extra antibodies, whatever is there, it has to be drained off, which is why 2 to 3 times you wash it so that the proper reaction takes place. Then chromogen solution is added to the wells, and again it is incubated. So we get a blue color after all these steps, and then a stop solution is added. The stop solution will change the color from blue to yellow, and then this particular plate is placed into the ELISA reader, and we will check for the absorbance. So based on the color of your final plate, we can make out whether the particular product is there or not, whatever we are looking for, and we get a graph onto the reader. Now, testing and method development. So quality assurance, as I mentioned, is extremely critical. So in the laboratories, testing is also done. Apart from that, we are all constantly innovating and developing different types of methods. So whatever testing is done in food laboratories, it has to ensure that the processes that are involved are accurate, and then whatever results are coming out, they have to be accurate and precise. There is repeatability, reproducibility, and the expertise that is involved. The hand that is doing the analysis has to be technically completely sound. So we as a laboratory at Envirocare have been doing this from past 40 years. And so these are some of the accreditations and approvals that we have. So I think I am done with the presentation. So I just want, like I just started with a thought, I would like to end it with a thought. So the problem is a way to success, but there is a most important connecting factor in between the solution, and that solution is you. So in the industry there are different types of problems. Whenever as students you can look into those problems and innovate and create something new, and it will definitely lead to success. So thank you, and these are my contact details. You can definitely connect with us.
Thank you so much, ma'am. Hello, Anuprita, ma'am.
Yeah, thank you.
Yeah. Thank you so much. It was a very informative session, ma'am. We can start with the question and answer session now. The first question is explain dot blot technique.
Dot plot technique is kind of like it is also on the same basis of antigen antibody interaction. Hello.
The next question is what is the difference between recombinant DNA and gene editing?
So, recombinant DNA technique is wherein we are creating something. We are going to introduce a particular gene of interest into the sequence, and this is used to produce BT cells. Whereas gene editing is different. Gene editing is where you alter an existing gene. We are not introducing anything different. We are editing the gene, and this is usually done with the help of CRISPR-Cas9 technology, which is the latest technology on gene editing.
Which advanced molecular markers is used nowadays for identification of adulteration in food product as there are RAPD, ISSR or RFLP?
So, like I mentioned in the meat speciation, the COX gene, that is it is a mitochondrial gene. This particular gene is a marker that is used, and this particular molecule can even differentiate between very small kinds of mutations that take place.
Which is the best technique to avoid food adulteration?
Avoid food adulteration.
Yeah.
There is no technique to avoid food adulteration. We can just assure that okay, that food adulteration is not taking place. So, as a laboratory or as a food business operator, also they need to get it tested on a regular basis, and based on whatever kits are available in the market, we can test for the products. And to avoid, of course, whoever is doing it has to stop doing it. Yeah.
Which PCR technique is currently used commonly in industries?
It is real-time PCR. Like I mentioned, commonly used technique is real-time PCR. It is extremely advanced and it is very sensitive also.
Minimum how many primers are needed to use for RAPD or ISSR PCR and how can we select that?
It depends on what exactly you are screening for. So all this information is usually given in the kits. So based on what exactly you are screening for, primers are basically if you are trying to look for one particular gene, so one particular primer is used that is specific for that gene. So there is a reverse primer and a forward primer for that. But if you are trying to multiplex it and try to screen for different genes, then accordingly the number of primers will vary.
Okay. Which molecular technique we can use for differentiation of turmeric samples from various region to differentiate at molecular level?
Turmeric samples, even I am not aware about it actually. I will just look into it and I'll get back.
Okay. Any other compound which is used instead of horseradish peroxidase in ELISA testing?
We can use alkaline phosphatase. So that is complex to the antibody. These enzymes are conjugated to the antibody and then the reaction takes place. So typically apart from horseradish peroxidase, alkaline phosphatase is also used.
Which standard GMO markers are used in food testing?
For GMO markers in food testing, the most common ones, as I explained, was CAMV35S promoter and Tinos terminator. This is the most common one. But apart from that, lately it has been seen that in cotton samples, we are also screening for four additional molecules. That is FMV, then cryogenes, uh, PAT, and one more EPSPS. These are the genes additionally that are screened for. But most common is CAMB and 35S.
Okay. What are the disadvantages of GMO?
Oh, so basically, it has not been proven anywhere, but the long going debate about GMO is that it can affect. So, if you go on planting GMO for a long period of time, it can have a negative impact on the soil quality. And also, we are producing GMOs in order to develop resistance to a particular pest. But it so happens that sometimes the pest become just like antibiotic resistance. Same way, even the pests become resistant to the GMO. So that is a big problem. And then how do we deal with this issue? So that is why it is a very big impact, negative impact on GMOs.
Okay. What if we have to test several samples at one time? So can multiplex PCR be used?
Yes, yes. Multiplexing is actually different, but when you want to run multiple samples at a go, PCR usually you can run many samples together. It is a 96 well plate, and there are even higher versions of it. You can run around 200 to 300 samples also sometimes together.
Okay.
Yeah.
Which enrichment media is used for pathogen testing?
Buffered first. Buffered pattern water. BPW.
Okay, ma'am. Thank you so much. I'll ask Aditi ma'am to offer the vote of thanks now.
Thank you, Swana. I would like to thank our young and dynamic speaker, Miss Anuprita Raichurkar, for giving us her valuable time and a wonderful webinar. Ma'am, you have beautifully explained these molecular biological techniques in an extremely simplified form, and I'm sure that the basics are now very clear to the students. I also would like to thank Envirocare Labs for supporting our department in conducting such a wonderful lecture on molecular biology. I would like to thank our principal, sir, Dr. Kiran Mangaonkar, for always encouraging and supporting us. I would like to thank our HOD, Gauri ma'am, for being the guiding force throughout the webinar, and would also like to extend my gratitude to my colleagues Subhashini ma'am and Suhana for looking after the entire proceedings. Also, this webinar would be incomplete without the technical support of our IT team. Aldrin sir, Sham sir, Ashish sir, thank you so much. And lastly, I would like to thank all the participants for such an overwhelming response. We are also coming up with few more webinars on microbiological testing for food products and their permissible limits. And the second one is the accelerated shelf life studies, both of which details will be posting shortly. One more small announcement regarding the certificates as well as the feedback link. The feedback link has been provided in the chat box. So, all the participants who are attending the meeting can fill in the feedback and send it to the respective mail IDs. Also, I would like to inform you that the because of the overwhelming response, we have a high number of participations, and that is why it will take around 10 to 15 days, maximum 15 days for the certificates to reach the candidate. So you are requested to kindly be patient with us. Thank you, Anuprita ma'am, once again for this valuable lecture. I'm sure our students have benefited. Thank you so much, ma'am.
Thank you. Thank you so much for having me here.
Yes, thank you, Anuprita.